Hello everyone, welcome back to the channel. In the previous video, we have discussed about linkage. What is linkage? What do you mean by linked gene? What is unlinked gene? How to calculate the recombinant frequency? All that we have discussed in the last part of the video. Today we are going to continue this with the next part, that is the gene mapping. Okay. What is gene mapping? Gene mapping is nothing but to find the location of the linked gene in a chromosome. Okay, as you know, mapping, generally if you see a map, world map, what is the role of world map? It states or it shows where the countries are present or where the states or specifically locations of each area. That's the role of mapping. Similarly, gene mapping is related to the location of the gene on a chromosome. To be very specific, the location of the linked gene on the chromosome is known as gene mapping. The gene mapping can be otherwise told as linked gene mapping or chromosomal mapping. Why it is called linked gene mapping? Because we are finding the location of the linked genes, and this is called chromosomal mapping because we are finding the location of the genes on the chromosome. That's why the gene mapping is called so. And what are the main purposes of gene mapping? Why are we considering gene mapping so important? We will discuss in this part of the video. So let's begin. Before that, let's understand the definition of gene mapping. The linkage of the genes in a chromosome can be represented in a form of genetic map called as the linkage map or chromosomal map. This definition I have already discussed. Next, what are the two aspects or why we are doing gene mapping? There must be certain purpose or certain function, or you can say certain properties of gene mapping. Gene mapping is done only to find two things, that is gene order, second is genetic distance, or you can say gene distance. What is gene order and what is gene distance we will be discussing in the later part. First, we need to understand what are the unit to be measured for the gene map. As you measure the length by cm, here the measurement of genetic map is centimorgan. It is denoted by cm, but it is small c and capital M. Okay. So, this unit of measurement of map unit is called centimorgan. One map unit is equal to 1 cm, which is equal to 1% of recombinant. Okay. Now you must be clear why we are going to find the genetic distance or genetic mapping. Because this genetic mapping shows the distance, not only the gene order, but also the recombinant frequency. How we can find the recombinant frequency by this? Because 1 cm or 1 centimorgan is equal to 1% frequency. Let's say if you get 10 cm, then the genetic frequency or the recombinant frequency will be 10%. There is both way you are getting benefited. By knowing the distance, you are knowing the recombinant frequency, genetic order as well as gene distance. So three things can be done through the genetic mapping. That's why genetic mapping is very, very important. Here is a very simple formula for the map distance, which we have already discussed in the previous video regarding recombinant frequency. Here, the map distance is equal to number of recombinant offspring by total number of offspring into 100. Sorry, this has been moved upward, but you can write it right here. Okay, wait. Okay, like this. Number of recombinant offspring by total number of offspring into 100. So this formula we have already discussed regarding when we were discussing about the recombinant frequency. The same formula is used to discuss to calculate the map distance. Okay. Next, moving on to the example so that you get a clear picture how to calculate the gene map or the distance. So let's begin. Suppose the two parental combination are given, that is capital A, capital B and capital B and small a, small b, small b. When you make a cross, you get this F1 progeny, as this is a normal Mendelian cross, and then in the F2 generation you get this following progeny. This following progeny have this following number, that is capital A, capital B, small a has 160 offspring, this is 165, then 36, then 39. So you get total of how many offspring? Of 1000 offspring. Okay. What you do? You need to first find the parental and the recombinant one, which we have already discussed in the previous video, how to find which offspring is parental, which is recombinant. The trick is you need to see the highest number among this four. Among this four, the highest number are 160 and 165. So the parental offsprings are these two. Okay, and the recombinant one will be the least one, that is 36 and 39. And now your problem is almost solved. You need to just put the formula and find out the distance. How you will find out the distance? Normal formula, that is map distance equal to number of recombinant offspring. Here the number of recombinant offspring is 36 plus 39 divided by total number of offspring, means you need to add up 160, 165, 36 and 39 into 100. So you get the answer, that is 18.8 cm. So you can see here the genetic distance between A and B is 18.8 cm. And by this you can also know the frequency of this generation, that is 18.8%. Just you need to just convert into percent. 18% is the recombinant frequency and 18.8 is the distance between the gene. By this you just need to know what is parental combination, which is recombinant combination, and you need to just put the formula. By this you can calculate the gene distance or gene map. Next is the gene order. What is gene order? Map distance between the two genes are allowed to place genes in the proper linear order. Let's say there are three genes. Suppose A, B and C. In order to know that whether first of all A is present, then B is present, or C, or arrangement is A, C, B, you need to know the first the gene order. In order to calculate the gene order, you need to first do certain thing. We will understand this concept regarding one example so that you get to understand this better. Suppose this is an example, and here you are given certain phenotype as well as the number, and you need to find out which one is parental and which one is double cross. What is double cross, single cross, we will be discussing in the later. After some time we will going to discuss that. But first you need to find out which one is parental. The only trick is the largest number is the parental combination and the least one is the double cross. Okay, this much you need to understand here. Later on I will be discussing what is double cross and single cross. First we will find the gene order. So what you need to do, first you need to find the parental and then the double crossover. Now you need to compare this both. What you will do, this C here. BAC, capital BAC is the parental type 1. Sorry, capital BAC is the parental type 1 and small BAC is the parental type 2. Okay, and BAC, small, this is small c, for very sorry, this capital is written, but this is small c. Okay, and capital B, small a, small c is the double crossover 1, and small b, capital A, capital C is the double crossover 2. Now what you need to do, you will compare parental type 1 with the double crossover 1 and next parental type 2 with the double crossover 2. See, parental type 1 with the double crossover 1 you need to compare, and wherever the change you will find, here capital B same, small c is same, but the change occurs here, that is capital A, small a. Here also if you compare the parental type 2 and double crossover 2, you will find the change right here. The trick is that wherever the change is present, the change should be present in the middle. Change means that is the difference. You will find the difference only in the middle. Here as the change is found in the middle only, you no need to change the order. The gene order will be as it is, that is BAC. Next example you will get clear idea. See, this example is better. Suppose the parental genotype is this, that is capital A, capital B, capital C and small a, small b, small c, and the double crossover you need to find with the least number, that is capital A, capital B, small c and small a, small b, capital C. What you need to do? You need to compare the parental type 1 with the double crossover 1 and parental type 2 with the double crossover 2. See what you have done? Here you will see that capital A, capital B, capital A, capital B both are same. But the difference occurs here, that is capital C, small c and also here also capital C, small c. But remember one thing, the change should always occur in the middle. As this change is present in the corner side or in the extreme left side, you need to just arrange this C or the change in the middle and you get the gene order. As the C is present right here in the corner, you just need to add right in the middle and your gene order will be ACB. But in the previous one, you have seen that the difference was present in the middle only. The change was present in the middle only. You need not have to change any order. The order will be same, that is BAC. But as this case, the change was present in the extreme left, you need to just write in the middle and you get the answer. That is the gene order. Okay. Why this change is occurring in the middle is due to double crossover and we will find it in this upcoming class. Okay. Now we will understand what is 2-point cross and what is 3-point cross. If the cross involves two loci means two genes, that is A and B, then it is known as 2-point cross. And to find 2-point cross, the formula is very simple. Same formula, number of recombinant by total number of progeny into 100. Here you need to just find the parental combination and the recombinant combination as I have already mentioned, and you need to put this formula and you will get the result. Okay. 2-point cross is very simple, which we have discussed earlier also. Main coming to the 3-point cross. In this 3-point cross, you will understand three things. First is what is single crossover 1, single crossover 2, double crossover. What is 3-point cross? Cross involving three loci. Here you can see three loci are present, that is A, B and C, and this is known as 3-point crossover. First you will understand what is single crossover 1. If the crossover takes place first position, that is once or first in the first place, that is between A and B, then this is known as single crossover 1. See, here the crossover takes place between A and B. If you make a crossover, what you will get the progeny? Capital A, as the crossover begins here from capital A, small b, you can see here capital A, small b and small a, capital B, small a, capital B. This C will remain as it is. There will be no change in the C. Okay. So here capital A, capital B, small, sorry, there will be change. Capital A, capital B, small c. You can see here. And here you will write from here side capital A, capital B, capital C. Okay. Only the cross between A and B was made. Then this is single crossover 1. If you make the crossover between B and C, this is called single crossover 2. See, from capital B, capital C, okay, crossover, and capital C, small b crossover. This is known as the single crossover 2. And what will be the progeny? Capital A, capital B, and here the turning will be placed small c. Similarly, here also small a, small b, and here it is placed, that is capital C. Okay, wherever the cross will be present, you need to just interchange that position only. Here also you need to interchange the position only wherever the cross is present. Okay, so this is single crossover 1 and this is single crossover 2. Hope you understood that how we got the progeny. Okay, because this is very important. Next comes to the double crossover. If you make the cross between both this place, then it is known as double crossover. Between A and B also, B and C also. How you will get the progeny? Starting from A, then moving to small b, then here again a cross is present, you move to capital C. See, capital A, small b, capital C. Okay, next again small a, cross is present, capital B, again cross is present, small c. Okay, small a, capital B, small c. So you get the progeny like this. And you can see here that in the parental type, the capital A small a and capital C small c remains same in the progeny. But only change occurs here in this middle position. Okay, you can see here also. The capital B was present upside, small b was present downside. But here the change only occurred in this position. Rest, both the extreme ends remains the same. That's why when you find the gene order, comparing the parental type and the double crossover, the change always occurs in the middle. If the change is not present in the middle, then you need to just change the place and you can find the gene order. Now you must be clear why this gene order, the change was always present in the middle. This is about the 3-point cross. Let's take an example so that we get a clear picture. Next we get to find, first we need to find the parental. How we will find the parental? That is by observing the highest value. Here the highest value is 390 and 374 is the parental. And the least value will be double crossover. Okay. Then you need to find the gene order. Here the gene order will be ACB. Okay. If you compare the parental 1 with double crossover 1 and parental 2 with double crossover 2, you get the gene order, that is ACB. Next you need to do a single cross between AC and you need to find the progeny and compare this with this table and you will find the progeny between A and C. Single crossover between A and C. Next again you need to do a single crossover between C and B. You will get progeny and you will compare this with this table as we have done here. See, here single crossover is done, progeny we get. This progeny you need to compare with the table where you are getting this type of progeny. That will be a single crossover 1. Second, again you will do the single crossover 2 and you need to compare this progeny with the table and you will know the single crossover 2. If you want to know a trick how you can find the single crossover 1 and single crossover 2, you need to see this. First you will find the parental. Second, the double crossover will be the least value. After parental, the next parental and double crossover, you need to find the gene order. Suppose the gene order is ACB. So which one will be the single crossover 1? AC. Single crossover 2 will be CB. So AC, that is the number of progeny or the number will be after parental, the next highest number will be of single crossover 1. That is between A and C. And then after that, single crossover 2 will be the next number. Okay, next highest number, that is 27 and 30. And the least value will be 5 and 8. So you see the first greatest highest number, that is parental, will be that 2, and find out the least number, that is double crossover. Next you need to find the gene order. After finding the gene order, you will get, suppose ACB. Then AC is the single crossover 1 and BC is the single crossover 2. Then the AC value will be, the single crossover AC value will be just below the value. Means after parental, next highest value will be 81, 85. Okay, you compare this, you can understand it better. Next, after that, below that you will get 27, 30. That is single crossover 2 will be the next highest number. And the least number will be 5 and 8. So by this you can easily within few seconds know the type of gamete. Once you get this type of gamete, because in exam only genotype and observed value will be given, you need to find out which one is parental, which one is double cross, which one is single cross, which is single crossover 1 and 2. For that I have given you this simple trick. Okay, and if you want to cross check, you can also cross check by crossing singly as well as doubly with the parental combination, so that you get sure that your combination or the type of gamete which you have written is correct or not. Okay, next what you will need to do, after finding the gene order, you will find the distance between A and C. So to find the distance between A and C, you need to add single crossover 1, that is 81, 85, plus you need to add the double crossover also, because whether you are taking single crossover or single crossover 1 or 2, you need to add the double crossover. Okay, divided by total number of progeny, as the formula says, into 100, we get the result, that is 17.9. Okay, we get the result, that is 17.9 distance between A and C. Next, similar C and B, that is 27 and 30, that is single crossover 2 plus 5 plus 8 divided by total number of progeny into 100, you get 7 cm. That is the distance between C and B, and distance between A and C is 7.9 cm. By this you must be clear that how to calculate the gene mapping. First of all, you need to locate which is parental, which is double cross. Okay, first thing. Second, you need to find the gene order. Okay, after finding the gene order, you need to find the single crossover 1 and single crossover 2. By crossing you can find out, otherwise you can just find the next highest number of the parental will be the single crossover 1, below that will be single crossover 2, and the least will be double crossover. Then you need to just put the formula and you can find the genetic distance between them. So this is all about the gene mapping. Okay, hope you are clear with the idea what is gene order, what is gene mapping, and how you need to calculate it. So that's all for today's video. Hope you have a very fruitful session, and if you have any doubt regarding this, you need to just ask in the comment section and share among your friends as much as you can, because this is very important concept, that is linkage, genetic map. The genetics questions are usually asked from this concept only, from this topic only. So if you are not clear, you need to just clarify your doubts by watching this video over and over again. Hope you will get clear with this, and like, share and subscribe to my channel if you are new. Thank you and have a nice day. That's all for today.